Psilocybe cubensis is a species of psychedelic mushroom whose principal active compounds are psilocybin and psilocin. Commonly called shrooms, magic mushrooms, golden halos, cubes, or gold caps, it belongs to the fungus family Hymenogastrophaceae and was previously known as Stropharia cubensis. It is the most well-known psilocybin mushroom due to its wide distribution and ease of cultivation. Taxonomy and naming. The name Psilocybe is derived from the ancient Greek roots psilos and Cuban translates as bear head. Cubensis means coming from Cuba, and refers to the type locality published by Earl. Sanger divided P. cubensis into three varieties: the nominate, which usually had a brownish cap, Morellus cyanescens from Florida, which generally had a pale cap, and var cerulescens from Indochina with a more yellowish cap. Psilocybe cubensis is commonly known as gold top, golden top or gold cap in Australia, sacred mushroom or blue mushroom in Brazil, and San Isidro or palenque mushroom in the United States and Mexico, while the term magic mushroom has been applied to hallucinogenic mushrooms in general. A common name in Thai is head kai, which translates as mushroom which appears after water buffalo defecates.
The cap is 1.6 to 8 cm (0.6 to 3.1 in), conic to convex with a central papilla when young, becoming broadly convex to plane with age, retaining a slight umbo sometimes surrounded by a ring-shaped depression. The cap surface is smooth and sticky, sometimes with white universal veil remnants attached. The cap is brown, becoming paler to almost white at the margin, and fades to more golden brown or yellowish with age. When bruised, all parts of the mushroom stain blue. The narrow gray gills are adnate to adnext, sometimes succeeding attachment, and darken to purplish, black and somewhat mottled with age. The gill edges remain whitish. The hollow white stipe is 4 to 15 cm (2 to 6 in) high by 0.4 to 1.4 cm (0.2 to 0.6 in) thick, becoming yellowish in age. The well-developed veil leaves a persistent white membranous ring whose surface usually becomes the same color as the gills because of falling spores. The mushroom has no odor, and tastes farinaceous. The spores are 11.5 to 17.3 x 8 to 11.5 micrometers, subellipsoid, basidia 4 spored but sometimes 2 or 3, pleurocystidia and chylocystidia present. The related species Psilocybe subcubensis, found in tropical regions, is indistinguishable but has smaller spores. Distribution and habitat. Psilocybe cubensis is a pan-tropical species, occurring in the Gulf Coast states and southeastern United States, Mexico, in the Central American countries of Belize, Costa Rica, and Guatemala, the Caribbean countries Cuba, the Dominican Republic, Guadalupe, Martinique, and Trinidad, in the South American countries of Argentina, Bolivia, Brazil, Colombia, French Guiana, Paraguay, Uruguay and Peru, Southeast Asia, including Thailand, Vietnam, Cambodia and Malaysia, India, Australia including Tasmania, New Zealand, Fiji, and possibly Nepal and Hawaii. Psilocybe cubensis is found on cow and occasionally horse dung, sugarcane mulch or rich pasture soil, with mushrooms appearing from February to December in the Northern Hemisphere and November to April in the Southern Hemisphere. In Asia, the species grows on water buffalo dung. Along with other fungi that grow on cow dung, Psilocybe cubensis is thought to have colonized Australia with the introduction of cattle there, 1,800 of which were on the Australian mainland by 1803, having been transported there from the Cape of Good Hope, Kolkata and the American West Coast. In Australia, the species grows between northern Queensland to southern New South Wales. In March 2018, several Psilocybe cubensis specimens were collected in Zimbabwe in the Wedza district of Mashonaland East Province, approximately 120 kilometers southeast of Harare. This was the first reported occurrence of a psilocybin mushroom in Zimbabwe. The mushrooms were collected on Imire Rhino and Wildlife Conservation, a nature reserve that is home to both wildlife and cattle, as well as cattle egrets. Psychedelic and entheogenic use. Singer noted that Psilocybe cubensis had psychoactive properties in 1949. In 1996, jars of honey containing Psilocybe cubensis were confiscated at the Dutch-German border. Upon examination, it was revealed that jars of honey containing psychedelic mushrooms were being sold at Dutch coffee shops. Psilocybe. Cubensis is probably the most widely known of the psilocybin-containing mushrooms used for triggering psychedelic experiences after ingestion. Its major psychoactive compounds are psilocybin, psilocin, biocystin, norbiocystin. The concentrations of psilocin and psilocybin, as determined by high-performance liquid chromatography, are in the range of 0.14 to 0.42% and 0.37 to 1.30% dry weight in the whole mushroom, 0.17 to 0.78% and 0.44 to 1.35% in the cap, and 0.09 and 0.30%, 0.05–1.27% in the stem, respectively. Individual brain chemistry and psychological predisposition play a significant role in determining appropriate doses. For a modest psychedelic effect, a minimum of 1 gram of dried Psilocybe cubensis mushrooms is ingested orally. 0.25 to 1 gram is usually sufficient to produce a mild effect. 1–2.5 grams usually provides a moderate effect, and 2.5 grams and higher usually produces strong effects. For most people, 3.5 dried grams (1/8 of an ounce) would be considered a high dose and may produce an intense experience. This is, however, typically considered a standard dose among recreational users. For many individuals, doses above 3 grams may be overwhelming. For a few rare people, doses as small as 0.25 gram can produce full-blown effects normally associated with very high doses. For most people, however, that dose level would result in virtually no effects. Due to factors such as age and storage method, the psilocybin content of a given sample of mushrooms will vary. Effects usually start after approximately 20 to 60 minutes, depending on method of ingestion and stomach contents, and may last from 4 to 10 hours, depending on dosage. Visual distortions often occur, including walls that seem to breathe, a vivid enhancement of colors and the animation of organic shapes. The effects of very high doses can be overwhelming depending on the particular phenotype of cubensis, grow method, and the individual. It is recommended not to eat wild mushrooms without properly identifying them, as they may be poisonous. Cultivation. Psilocybe cubensis grows naturally in tropical and subtropical conditions, often near cattle due to the ideal conditions they provide for the growth of the fungus. The cow usually consumes grains or grass covered with the spores of Psilocybe cubensis, and the fungus will begin to germinate within the dung. Mushrooms such as Psilocybe cubensis are easy to cultivate indoors and grow on numerous substrates such as coconut coir, coffee grounds, brown rice flour, sawdust, and straw. Rye grain is said to be the most suitable substrate for growing Psilocybe cubensis. While growing mushrooms inside a jar, it is recommended to leave the container at room temperature or case it with soil. A study conducted in 2009 showed that mushrooms grown in the dark had higher levels of psilocybin and psilocin compared to the mushrooms grown in bright, indirect light, which had minimum levels. Studies were conducted where an environmentally controlled wind tunnel and a computer program were used to determine the influence of humidity on the individual basidiocarps of Psilocybe cubensis, which aided in mapping their growth and development. The transpiration and growth of the mushroom was heavily influenced due to the humidity of the air, and the transpiration was accelerated at higher humidities, while light had no effect to the growth. Faster growth was observed at higher humidities. It was also discovered that misting enhanced both the growth and transpiration rates in the growing process of Psilocybe cubensis. Mushrooms grown with PF tech. Personal scale cultivation of Psilocybe cubensis mushrooms ranges from the relatively simple and small-scale PF tech and other cake methods that produce a limited amount of mushrooms, to advanced techniques utilizing methods of professional mushroom cultivators. These advanced methods require a greater investment of time, money, and knowledge, but reward the diligent cultivator with far larger and much more consistent harvests.